Hello everyone, myself Supratik Kaur. I'm a assistant professor in the Department of Chemistry and Physics. For the research day 2025, I'm presenting my research titled PDLI version 1, an open source web-based tool for predicting drug-induced liver injury, or DILI. Predicting drug toxicity is crucial for minimizing adverse effects, reducing late-stage failure, and improving drug safety. Computer-aided drug design and machine learning model enhances early detection by analyzing vast chemical datasets, identifying toxic structural patterns, and streamlining risk assessment, ultimately accelerating safer drug development throughout the time. Here you can see the possible drug toxicity we checked when we design a new drug through the computational chemistry. In the present study, we are targeting drug-induced liver toxicity, which is known as hepatotoxicity. This is the complete scheme of the present study where we took a drug-induced liver injury datasets of 1,159 drugs, where more than 50% compounds are proven DILI toxic, and other remaining ones are DILI non-toxic. We divided the dataset into training and test sets in almost 3:1 ratio. We analyzed the chemical space, followed by the feature extraction and the feature selections, and then we developed models through the random forest, and followed by whatever the model we got it, we employ that model in the Streamlit, which is a Python, which is a web-based application. Whatever the Python code we prepared, that specific code as well as the model we employed in the Streamlit. In this present slide, you can see that representative drug compounds with the DILI toxic and DILI non-toxic fingerprints and the substructural features. DILI toxic fragments promote DILI risk, while DILI non-toxic fragments hinder DILI risk. So if you see the substructures or the toxic fingerprints, the blue ones are the toxic fingerprints. So if you see a drug which has any of those blue fingerprints or the structural scaffolds, technically you can think that the drug can be DILI toxic. On the other side, the yellow ones, whatever the fragments you can see, if you see those fragments are present in your drug, that means they are non-toxic fragments, or the fingerprints which are good for your drug not to have the drug. Specific figure illustrates how S-log-P and Gas2D influence the binary classification outcomes, such as predicting a molecule's toxicity, for example, DILI risk versus non-toxicity, highlighting their roles in the structural property relationship. Now, if you see the S-log-P, which is a logarithm of the octanol water partition coefficient, quantifies a compound's partition between octanol and water, calculated via the atomic contribution method and reflecting mostly hydrophobicity or your lipophilic property. On the other side, if you see AAT-S0V, which is an average atom type electrotopological descriptor, is a valence-based descriptor averaged from electrotopological indices focusing on the immediate atomic environment. Now, depending on their pattern, you can see the more on the yellow color zone, if a drug is existing, they are mostly toxic or class 1. On the other side, the more it's going towards the indigo color or towards the minus section of the S-log-P, on the other side, around the range of 180 to 200 for the AAT-S0V, you can see the drugs are becoming more non-toxic. That's a very, very important observation when you will use this specific model for predicting a new drugs. And those two values can give you an idea that what kind of toxicity features you can see for the DILI, if the drug is DILI toxic or non-toxic. To make this model user-friendly or available open access for any users who are interested in drug design and drug discovery, we have used our or we have deployed our model as well as our script in a Python-based web application in the Streamlit. You can see the link there, and you can see the screenshot of our web server. Along with that, we have prepared two more options. One is the code is available in the GitHub, and one can use this code for their prediction in the Google Colab notebook, or one can use as a graphical user interface with the Anaconda environment. Now, if you see the web app PDILI, once you click the link which I mentioned in the previous slide, you can see the specific first screenshot. And once you know your drug smiles, you just need to put the smiles. As you can see, the smiles of sorbitol is provided here. The moment you will enter it, you will see the structure, you will see the prediction, is it a risky to the DILI or it's a non-risky, as well as it will show you that if the prediction is following the applicable domain, which is one of the most important OECD principle. And if it's a yes, that means your prediction is absolutely reliable. You can also see your compounds, which is shown in green, our molecule, that which is within the acceptable AD limit. Interestingly, this specific work just accepted on 18th of March 2025 in American Chemical Society ACS Omega Journal, which is a Q2 journal with an impact factor of 3.6. To acknowledge, I want to acknowledge my collaborator SK Abdul Amin and Stefano Piotto from the University of Salerno, Italy. And I want to acknowledge President Lamont Rapale, Provost David Bursell, Dean George Chang, and Chair Professor Heather Stokes-Hooby of Chemistry and Physics for the research release time and the computational resources. Hope you all had a great research day. Thank you so much.